All right, welcome back. Here we're going to talk about the history of microbiology. In the beginning of microbiology, we start with the cell theory in 1665 with Robert Hooke. He's the one that came up with cell theory, and cell theory has three parts. You do need to know the parts of cell theory. They consist of that all things are composed of cells. All cells come from other cells, and that the cell is the smallest unit of life. At this point, a lot of this was just theory and hadn't really been proven, so it wasn't necessarily accepted at this point. But Robert Hooke was the first one to propose this. Then in 1973, microbes were actually observed for the first time by Leeuwenhoek. As I mentioned, this wasn't necessarily accepted because a lot of people did believe Aristotle's doctrine of spontaneous generation. This was quite a bit debate for quite some time because a lot of people at the time believed that living organisms could arise from non-living matter. It was said that it was a vital force that formed life. This was in opposition to biogenesis, which is the theory that living organisms arise only from other pre-existing life through reproduction. So we're going to talk about a series of experiments done by several different guys where they were trying to prove either spontaneous generation or biogenesis. So in this first experiment, by Reddy in 1668. He took two different jars, or two different types of jars. He did three of them that were covered, and that prevented the flies from entering to get to the meat inside the jar. And then three of the jars were uncovered, which of course the flies could get in, and then this served as the control. And what he found was that in the jar that was uncovered, there were maggots. And in the jar that was covered, there was not maggots. And so this was to prove that biogenesis was true without prior life, which would represent the flies. Without the flies, then there were no maggots. And so that's what he was suggesting, that it required the flies to produce maggots. John Needham in 1745 then proved that microbes arose from fluids. So what he did is he took chicken broth or corn broth in flasks, and he would boil it and then put it in a flask. So it's important to note the order here. So he boiled broth and then put it in this flask and sealed it. And so what he found is that that would still lead to growth. So the point of boiling the broth was to kill off the microbes. But then he would put it in the flask and he would say, look, I killed all the microbes when I boiled the broth, and then I put it in this flask and it still grew. So no, we don't need prior life. Life can spontaneously generate. So this was evidence for spontaneous generation. But then along came this guy in 1764, Spallanzani, and he basically did the same thing, but what he did is he put the broth in the flask first and then boiled it. And so this was boiling the broth and heating the flask to make sure that the flask was sterile as well. And what he found was that no, we didn't have microbial growth. And so this was more evidence back for biogenesis. We know now that Needham, sorry, I just realized this isn't full screen. There you go. We know now that Needham did not sterilize his flask, right? So just because he sterilized the broth, when he poured that broth into an unsterile flask, the microbes that were in the flask then grew. But anyway, you kind of get the story where we have some evidence for biogenesis, some evidence for spontaneous generation. And then along comes Pasteur in 1861. And he demonstrated that microorganisms were present in the air. So that was one thing that he did. The second thing that he did is he was kind of the final experiment to prove that, yeah, biogenesis is truly, the prevalent theory. So what he did is, in the first experiment, he took nutrient broth, placed it in a flask. Both of them, he put it in the flask and then heated them. Some of them he sealed, some of them he did not seal. In the ones that were not sealed, microbial growth occurred. In the ones that were sealed, there was no microbial growth. So this is showing that we are killing all the microbes on the flask and in the broth. But if we don't seal it, then microbes can enter into the flask through the air. And so he did this final experiment. This is a really important experiment because it was vital for aseptic technique, which is something that we use a lot today. It's very important. So I do want you guys to know about this experiment. So what he did is he took this flask with the broth in, and then he bent this S-shaped neck and then boiled the broth. He boiled the broth inside of this flask here. And what that did is it sterilized the broth and sterilized the flask, killing all the microbes. It left, though, this flask open to the air to prove that it wasn't because life didn't exist inside. Like some people could be saying, life didn't exist inside because it needed oxygen. So he's disproving that, look, it has access to oxygen, but microbes, while they can simply fall into the flask, they don't make this S shape. They don't travel up through this S-shaped neck. And so what he found was that they were sterile and they stayed sterile, and there was no more microbial growth. This is why it's really important when we're doing aseptic technique, and we'll see once we get into the lab that we do need to keep the lid over our plate. So if we have a Petri plate here with our lid, we don't just open it. We make sure that we keep the lid on top to prevent any microbes that are falling from reaching inside of our Petri plate. You'll see that more once we get to aseptic technique. So now we know biogenesis, life comes from pre-existing life, comes from other cells. Now we're going to be moving on, talking a little bit about vaccination here. So the first vaccination in Western society was produced by Jenner, and it was the smallpox vaccination. It was huge because smallpox was very prevalent at the time. But we know that vaccination was actually practiced even earlier than that by the Chinese in the 1500s. Oh, I guess that's it. Okay. Pasteur, we already mentioned, he had that really important experiment proving biogenesis. He is considered the father of microbiology. And so it's starting with Louis Pasteur that we see this golden age of microbiology, this explosion of learning and study in microbiology. Some things that Louis Pasteur did. We already talked about disproving spontaneous generation. He also looked at wine fermentation. So fermentation is carried out by yeast, and through this process, they produce alcohol. So he studied that process, and he also studied wine spoilage, and wine is spoiled from bacteria. We already talked about in our last part of this chapter earlier, how yeast, remember that's a fungus, a bacteria is a prokaryote, right? So the bacteria are producing vinegar in the presence of air, and that's what's actually spoiling the wine. So this is something that Louis Pasteur found. So it was important to keep the wine away from the air so that bacteria could not spoil the wine. And also another big one from Louis Pasteur was pasteurization. So that's heating liquids very quickly to kill any harmful bacteria in them, like our milk. All right, so germ theory, kind of leading up to that point. So we have this guy, Semmelweis, and he was the first to advocate hand washing to prevent the transmission of disease. Prior to this time, it wasn't really understood that microbes cause disease. So if we don't understand that microbes cause disease, then you don't really see a need to be washing your hands or sterilizing your surgical instruments, or sterilizing surgical sites or anything like that. It was previously thought that disease was just caused by like misfortune or like karma or that kind of stuff. So he was the first one that said, hey, look, we know the microbes exist at this point, and maybe that's what's causing disease. So if we start washing our hands, I think he was the one that was working with childbirth. So he started advocating washing hands from delivering one child into the next, and that increased or decreased the mortality rate in those women tremendously. Following his findings, then we have Joseph Lister in the 1860s, and he was the first to advocate for antiseptic surgery. So what he did is he actually used phenol. There's a picture of him using phenol here, which kills bacteria. And so they would spray that phenol on the surgical wounds to sterilize them. Lister is where we get Listerine from too. Robert Koch. So he worked with anthrax. He ultimately was the one to prove the germ theory of disease. We're going to talk more about that germ theory of disease as well as his Koch postulates later in chapter 14. But he was very obviously important with those developments. He also developed the pure culture technique. We'll talk about that in lab. Paul Ehrlich, he looked at antibodies and how they were important in the immune response. But another significant thing that he did was he developed salvarsan, and that was a chemotherapy, the first chemotherapy drug. And I want to take a moment to say here, chemotherapy, I think a lot of us think of like cancer treatment. For microbiology, I don't want you to think that. Okay, so try to take that understanding, take it out of your mind, because in microbiology, when we're talking about chemotherapy, we're just talking about a chemical drug. Just break the word down. Chemo, chemical, therapy. So drug is like a therapy, okay? So any kind of drug that's a chemical is a chemotherapy. So this salvarsan was the first chemotherapy used to treat bacteria. In this case, we were treating syphilis. So here again, chemotherapy, the treatment of disease with chemicals. They can either be synthetic or antibiotics. Antibiotics is a chemical that is produced by a bacteria or fungi that inhibits or kills other microbes. Speaking of antibiotics, the very first antibiotic was penicillin that was discovered by Alexander Fleming. He actually discovered it accidentally, not looking for an antibiotic. What happened was he had a Petri plate, which he was growing bacteria on, something we're going to do in lab. And he got some contamination in it. He got some fungus contaminant in his Petri plate, something that we may do in lab as well on accident, but it happens. Fungus is everywhere, and I do see that quite often in our teaching labs. But what he noticed was when he went to throw away his plate because it was contaminated, which is something we may do in lab if our plates get contaminated. As he was throwing it away, he noticed that surrounding the fungus, so here's the penicillin is a fungus, okay? So surrounding the penicillin fungus, that there was this zone of inhibition here. So you note here where there's a limited amount of bacterial growth. All these colonies over here, this is bacterial growth, but we don't see it surrounding the fungus. And he noticed that. And he said, wow, I wonder if this fungus is producing something that's causing the inhibition of bacterial growth surrounding it. So then he started to investigate that. And that's how we got penicillin, the first antibiotic. It did take up until, so it was 1928 when he discovered this plate on accident. And it took it until the 1940s to be purified and go through clinical trials before it was readily available. All right, so that's history. Now we're going to kind of talk about some of the studies of today, and then we're going to wrap up by talking about some emerging diseases of today. So ology, study of, right? So bacteriology is the study of bacteria. Mycology, study of funguses. Parasitology is the study of protozoa and parasitic worms, or helminths, right? Remember that word. Virology is study of viruses. Today we have lots of technological advances, namely genome sequencing. So that's something that was recently developed. It's getting more and more efficient. And as it gets more and more efficient, it's used more and more often. So as we start to use genome sequencing more often, that's going to start to be used more for diagnosing and identification of microbes. And as it becomes more cost effective. Molecular biology is the study of how DNA directs protein synthesis. Genomics is the study of an organism's genes. And this genomics, as I said, is going to be more and more prevalently used for the classification of microorganisms. Recycling of vital elements. We kind of touched on this when we were talking about the benefits of microbes. So we know that microbes are very important in our ecological systems and in the environment. Bacteria convert carbon, oxygen, nitrogen, sulfur, and phosphorus into forms that are used by plants and animals. So we couldn't live without bacteria. Also, in our sewer systems, we use bacteria to help degrade that organic matter. We also use bacteria to help degrade and detoxify pollutants such as oil and mercury. So we do use bacteria a lot. We also use it specifically, this species here, Bacillus thuringiensis. That's a bacteria, though, that we use to kill insects. So instead of using chemical pesticides, we can use this Bacillus species to kill insects, and it's also not toxic towards people or pets. So I think it's mostly used for like slugs and worms, but it's a great alternative to chemical pesticides. All right, emerging infectious diseases. So a disease results when a pathogen invades hosts and overcomes the host's resistance. So resistance is resistance from the host, so us. So we provide resistance to diseases through our skin, mucous membranes, cilia, stomach acid, antimicrobial chemicals. So these are all ways that we try to ward off disease. But if a pathogen is able to overcome those barriers, then disease occurs. An emerging infectious disease is a disease that is new or changing or increasing, or has a probability of increasing in the near future. So these would be diseases that are currently new or increasing right now. So that's what we're going to talk about on the following slides, our current or recent outbreaks. So we probably heard of Zika virus. This is a big one right now. Spread through the bite of an infected Aedes mosquito. Now this is a species of mosquitoes. There's more than one species of mosquito. If you didn't know, there's quite a few. This particular species is particularly aggressive. So they do bite a lot. They actually can bite in the daytime as well. So you do need to protect yourself all the time, not just in the morning or evening hours. Also, Zika can be passed through intercourse. So if your partner has been bitten by a mosquito that had Zika, well, you can get it too. And it's still spread even if your partner or you don't have any symptoms, it can still be spread. And where it's really significant is that it can be spread from mother to fetus. And so what this causes is microcephaly. That's what we're seeing here, microcephaly. It's a small head. The head doesn't develop properly. It has impact on brain development. There's currently no vaccine or cure for Zika. So babies that are born with microcephaly, there's no treatment for them. So it's really important to be very careful during that time. Ebola, hemorrhagic fever. It's caused by the Ebola virus. It causes fever, hemorrhaging, and blood clotting. It's transmitted via contact with infected blood or body fluids. It was first identified near the Ebola River in the Congo. That's where it gets named from. There was a recent outbreak in 2014 in Guinea and DRC and killed thousands of people. MRSA stands for Methicillin-resistant Staphylococcus aureus. So this is a bacteria, and Staphylococcus aureus is actually a bacteria that you have on your skin all the time. But this is a certain strain of that species that is resistant to lots of different antibiotics, and it's particularly pathogenic. So it causes skin and systemic infections. We find it quite often in hospitals or areas where we have a lot of people close together or people with weakened immune systems. So we first saw penicillin resistance. So we saw the Staphylococcus aureus infections were resistant to penicillin in the 1950s. So they started using methicillin instead, another type of antibiotic. Well, they developed resistance to that in the 80s. So then they started using vancomycin. Then they developed resistance to vancomycin in the 90s. Staph aureus that is resistant to vancomycin is called vancomycin-resistant Staph aureus or versa. Influenza. So there's lots of different influenzas. H1N1 influenza was a big outbreak back in 2009 called the swine flu. That's what they called it. There was also the avian flu, H5N1. We're going to talk about what that means when we get to viruses. We saw that primarily infected birds, but then that spread to people as well. And of course, COVID-19. That's a pandemic we're in right now, currently, caused by the SARS-CoV-2 virus. It was first identified in December 2019. As of today, when I am recording this, December 18th, 2020, there has been 75 million confirmed human cases and 1.6 million deaths worldwide. So that concludes our lecture on chapter 1. I hope you enjoyed, and I'll see you guys soon for chapter 2.